Good afternoon, everyone. So I'll be talking about investigations of color coding, contrast adaptation, and vision encoding mechanisms in the chick retina. So the work was done in Professor Narayan's lab in JNC along with my senior Deepak and myself. So people who are unfamiliar, the retina is a thin slice of neural tissue. It's a transparent tissue that is found inside the back of your eye. So it's a pretty complicated system with there are 50 to 60 cell types in the retina alone, and there are supposed to be like 10 million photoreceptors and like 1 million ganglion cells which finally send the signal. So what happens is that the photoreceptor, there are rods and cones, they absorb the light of particular wavelengths, and then there is a lot of processing that is going on in this area with the horizontal cells, bipolar cells, and amacrine cells. And then after that, the process signal is sent to the ganglion cells, from which the signal is sent as the information is sent as spikes or action potentials to higher brain regions for further processing. So what we do in the lab is, so we take the chick retina is it has tetrachromic vision because it has 4 cones compared to us where we have only 3. And it is a daylight creature, and it relies highly on its vision for survival. So it's a highly interesting system to study with respect to the retina in itself. So the advantage of studying the retina is, like I mentioned before, the photoreceptors that are these cells, these are the only cells, roughly the only cells that can absorb light. And after the processing is done, and the ganglion cells, these are these cells, they send the information to the brain via axons. So the good thing is, the good thing for us is that only the ganglion cells are the cells which have any connection to the brain, and there is no feedback connection from the brain to the ganglion cells. So any information about the visual scene has to be gotten by the rest of the brain or the cortex from the firing patterns of these ganglion cells. And another cool thing is that the retina only sees light, and light can be given very easily. Pattern light or any sort of light can be given using LEDs or projectors or something like that. So basically what we can do is we can control the input, and we can look at the output via these microelectrode arrays. So these are these flat arrays which have so we work with a system which has 60 microelectrodes. So they can sample at 25,000 hertz, and they detect signals in the microvolt range, which is what the ganglion cell spiking would look like. So what we do is, so we give light, and because we are giving the light, we know what the stimulus is. We know the output of the ganglion cells because we are recording from them. So what we get is a nice input-output relation, and then so you can effectively see how these different types of, so as I said, the ganglion cells are supposed to be, there are 20-25 types, and all of them are supposed to carry separate information to the brain. And so we can maybe try to decode some of what these ganglion cells are doing. So this is, so I told you about the microelectrode array. So these are only, so I told we are 60 electrodes. One of them is a ground electrode, reference electrode. So there are 59 other electrodes for recording from in principle. So this is just like 4 electrodes from a recent recording. So this is a green light pulse of some intensity, which is given to all electrodes uniformly. I have just shown it on one electrode. So for reference, there is to 0 is when so 1 second the stimulus is given, but I have shown the earlier time to show that there is not much firing. But when you see when the light is switched on, there is firing on multiple electrodes. And even for such kind of a simple stimulus, you can see that the firing patterns are different. This one starts later than this. This one has more spikes than this. This one is different from this one. So all of these are different cells. So just for a simple full field flash. So what you do, so here also you can see so these spikes are from one neuron, and these spikes, the smaller ones you can see are clearly from a different neuron, because the signal strength is one of the big things is the closer you are to the electrode, the higher the signal will be on that electrode. So you have to separate out these cells which have there will be multi-unit activity on one single electrode because it's an extracellular recording, and thus you would have to separate out these units out to maybe just do some single unit analysis on this. So how do you do that? So in our lab, we work with a sorter called Spiking Circus. It's an open source Python sorter that is so that has been developed separately. So this is I hope it's not very small. So what you do is you take the data. So this itself is a bandpass filter data. It's 300 to 3000 hertz. So you take the data, bandpass filter it, do some spike detection, dimensional reduction by PCA, clustering, density estimation, and so it is a little bit of a it's a big process. So finally, what you get by doing all of this is you essentially get out these spikes. These are, so you get spikes of single neurons. And like you make sure that these spikes of the single neurons are of the single neuron and not noise or something like that. So that's what you finally get by this template matching process. So from the multi-unit activity, you've gotten the single unit activity out. So now, so as I said, RGCs are functionally diverse with some responding to increases in contrast. So contrast being the Weber contrast. So increases in contrast would mean they are like on cells, they increases in contrast, some to decreases in contrast, they're called off cells, and some to both, so they are called on-off cells. So the idea is that the full field flash is a very fairly simple stimuli. So if you want to get out these cells which are on selective versus off selective, so what we do is, so we first make the system go to a mean light level of some ambient background illumination. And then from there, what we do is we provide this is the source is an LED, and so we take random values from a Gaussian distribution, and we update so around the same mean that the system was adapted for, with a particular variance being the contrast range. So what we do is we start the stimulus, this is one section of the stimulus. So you can see, so this is the mean, and the values are taken of some variance around this mean, and this frame is updated every some 10 millisecond or 25 millisecond or something like that. So the idea is that like for seeing these contrast changes, there will be spiking activities of ganglion cells. And since you know the input that you have given because you have given the stimulus, and you know the output because you have recorded the output. So the idea is that for every spike, for a particular memory of the neuron, like you can assume that it's 500 millisecond, but you can just take it as 1 second also. You take a memory of the neuron, and what you do is you take the spike, take every frame before the spike, and so that is the stimulus, which is like a vector of over a long time period. So that elicited the spike is the assumption. And if you do that for all spikes that the ganglion cell that a particular ganglion cell has produced, that would mean that you would get the spike trigger average of the response of the cell. So what you would basically guess is if the cell is firing, you know that on average this is the stimulus that preceded the spiking. So basically this stimulus was given to so basically this is another fact. So this is light adaptation. So when you switch from a certain background illumination to higher background illumination, it starts at 0. You can see that the firing rate increases and then it goes down to a baseline value. And the same thing happens when like this stimuli is put. For example, you've put a background illumination, you start putting contrasts of different contrasts without, like when there was no contrast changes before, you change to high contrast stimuli. See here also you can see the same increases in firing. So this is not very surprising because contrast adaptation and light adaptation in the retina is supposed to happen in the bipolar layer. And thus, since they are both happening in the same place, maybe the kinetics of it is the same. So this is also something that we observed. So again, so coming back to the stimuli, so this is again like a 5-second section of the stimuli. So what we did is, so these are 3 different cells that we got from the spike sorting procedure. So these are histograms, post-stimulus time histograms that are recorded by doing 10 repeated trials of providing the same stimulus, and then like plotting the histograms. There you can see like, so the ganglion cell, this cell for example, it ignores the other changes in contrast, while it fires exactly for like a if you see from this is like a lot of negative contrast that you see here. So below here is the raster. So each line indicates a spike of this particular ganglion cell at the bottom. So this is the same event that is plotted, but the 10 events are plotted like behind. So this is 100 millisecond order. So you can see like the events are pretty precise, and they the time. So for the first spike, if you take the time, the error in the events, you will see it's in the order of 10 milliseconds, which is also seen in other mammals and other species. So this is the other idea that I was saying. So if you have so you have the spiking events, and then you take for every spike you take the stimulus before the spike, and then you do it for every spike, and then you average it out. So what you get is you get the spike trigger average, or what is the average stimuli that this cell sees before it fires. So here you can see, so these are some examples, some are obviously better than the others. See, this is a clear off cell. So this is the same cell that is shown here with the raster. So this across trials, it shows like very clear activity, and like you see that so what it responds to is basically like a long period of negative contrast in the on its receptive field / on its body. So this is another cell taken from the same recording, but you can see even though the same stimulus was given, the spike trigger average is different. So what this cell will fire for is different from what this cell will fire for. And this is again from another preparation, which is like these are like, so we are working on 2 colors, so this blue and green. So this is for another cell. So here also, so these off cells are some of the most numerous cells that we found in the retina. So one of the other things that we wanted to see is, so since we are providing green and so there is a color opponent theory that says that humans or mammals perceive color by opponent processes. That is, blue and yellow are opponent, and green and red are opponent. So that is why you will never see greenish red hues or reddish blue hues or something like that, because your eye does not work that way. So what we wanted to see is, so because the chick is a vertebrate, but the avian retina studies are very few, so we wanted to see how color perception is happening in the chick retina. So we wanted to see if there are like some color opponent cells, which, so what it means is that, so I will show you just, so the data is, so we have some early preliminary analysis. So what the color opponent cell means is that, so you will see here that this is the same cell A. So we have provided green light to it, and we have provided blue light to it, and we reverse calculate the spike trigger average for both of these colors. And what you see is that for at least some of these trials, and like, so there is like, so here you can see like it responds to positive contrast in the green, while for the same cell, if you provide blue, it responds for negative contrast. So this would make it, so the same is true for this cell, which is like it responds to negative contrast in the blue, while it responds to rather positive contrast in the green. So these are like these are one of the most numerously recorded cells that we found in the retina. So to summarize, so the spike trigger average, the protocol of doing the spike trigger average, the reverse correlation analysis, can be used to distinguish retinal ganglion cells, even spatially, even a full field flash spike triggered, I mean a full field flash stimuli. So another thing that we were able to do is like identify green on blue off cells, color opponent cells, which are found in the mammalian retina as well. So this shows that the chick retina is indeed may also, by like the other vertebrates, have the green on blue off, green off blue on color opponent circuits. So one of the other ideas is that by understanding the temporal / the coding nature of the retinal ganglion cells, we can develop a visual prosthetic or a visual aid that can maybe replace photoreceptors or something like that to emulate ganglion cell responses. Thank you.
Thank you for that excellent talk. Is there any question from the audience?
I just wanted to know, so you're recording this response to stimuli in the same neuron?
So basically, sorry if it was not made clear. So in the retina, these photoreceptors are the light absorbing cells. No other cells in the retina absorb light. Okay. So, but the photoreceptor itself does not directly contact the rest of the brain.
Okay, sorry, not neuron, my bad. Sorry, the same sample is being stimulated.
Yeah, stimulated and recorded. Yeah. So what you do is you sacrifice the animal, you place it on this electrode array, ganglion cell side down, so that you can record action potentials that spikes from it. And then what you do is, so the photoreceptors are there. You have not like torn the retina or something. The whole retina is there. So what you do is you just provide light. So the light hits the photoreceptor. It starts doing some processing. So we make sure that the retina is alive through and like functioning.
The question is, have you done experiments where you stimulated one first and the second one second, and then second first, and then the
you can't stimulate these cells. These cells do not have opsins. They do not absorb light.
No, I'm trying to understand if there's some kind of adaptation happening at the level of the receptors, then you make
adaptation happens. So that's why I also showed this figure, or maybe that was also not clear. So that's what. So so this is what happened. So there was no light before. Right. At 0 seconds, I switch on like some background uniform light. Okay. So what happens is that you see a very high increase in firing rate of one ganglion cell. This is one ganglion cell, but this was seen across the electrode array. And what you see is that the firing rate then goes down to a baseline value. Okay. Now, so if you were to provide some stimulus here of some changes in contrast, it may not be able to recognize it properly. But if you were to provide a change of contrast here, it would be able to do it, because it is
So you wait the required amount of time.
Yeah. So so the required amount of time I would assume is the time for this thing to like 1 by E of that, as in would be, I would assume that is the required amount.
Okay, I'll interrupt at this moment because we are running out of time. So while the next speaker sets up, yeah, you can carry on the conversation. Yeah. So is there any other question from the audience?